All right, listen closely. 9 out of 10 students taking the Cambridge Biology 9700 exam get cell structure questions wrong in exactly the same way. And honestly, it quietly costs them the grade they actually deserve. They walk out of the exam hall feeling super confident, completely unaware that a really simple, totally avoidable habit just tanked their score. I mean, I see it time and time again in the examiner reports. But today, we're going to fix that. Welcome to this explainer, where we're going to deconstruct the exact traps that examiners set and show you how you can leap straight into those top mark bands. So, let's dive right in and actually look at the trap itself.
The ER makes proteins. It has ribosomes on it. This is important for the cell. Now, does that sound familiar? It might even sound entirely correct to you at first glance, but this vague description right here, it's a massive examiner's red flag. It's what we call the classic cold open mistake. You know, you're describing basic biology, sure, but you aren't explaining the science in a way that Cambridge mark schemes can actually reward. Basically, you're leaving points on the table by writing general textbook summaries instead of applying absolute precision.
Now, let's contrast that flawed attempt with a genuinely top-band examiner response. Instead of just saying, the ER makes proteins, a top-scoring candidate writes something like this. The rough endoplasmic reticulum, covered in ADS ribosomes, is the site of protein synthesis. Do you see the difference there? It really isn't just about sounding smarter, it's about using precise terminology and establishing direct causal links. We specifically said rough ER, we explicitly called out ADS ribosomes, and we used the phrase site of protein synthesis. That right there is exactly how you secure the highest marks.
Section 1. Endoplasmic reticulum and ribosomes. Rebuilding the foundation.
Okay, so to make absolutely sure you never fall into that vague trap again, let's completely rebuild our foundational knowledge of the endoplasmic reticulum and ribosomes from the ground up. The absolute #1 rule from the principal examiner reports is a concept we call terminology initiation. Basically, you must explicitly define your terms before diving into the main biological mechanism. So never just write ribosomes. You need to define them as specific ADS ribosomes found in the cytoplasm and on the rough ER, or 7DS ribosomes found inside organelles like mitochondria and chloroplasts. Establishing this right off the bat tells the examiner you have exact, syllabus-specific knowledge. And the A-star blueprint for the endoplasmic reticulum really highlights this with what we call a logic loop. Top candidates, well, they don't just point out that the rough ER has ribosomes, they connect its physical structure directly to its function. They talk about the extensive surface area of the rough ER to explain exactly how it facilitates the high-volume protein production required for secretion. So the absolute crucial takeaway here is that you have to explain the why. You literally link the evidence, like the ADS ribosomes and the large surface area, directly to the concept of high-volume synthesis.
Now, when this builds into data questions, that distinction matters even more. We've got that clear split again, ADS in the cytoplasm, 7DS in organelles. But here is a major warning, the missing unit error. When you get a data question about these structures, examiners report that students frequently just forget to include their units. They'll write something like 12 instead of 12 micrometers, or they totally fail to convert millimeters to micrometers, and that is an immediate mark loss, no way around it. You absolutely must extract specific values from the provided tables and double-check those units every single time.
Section 2. The plant vacuole and tonoplast. Escaping the vague trap.
Right, now that we've got the ER down, let's tackle another major mark earner where ordinary revision honestly tends to fail us. The large, permanent plant vacuole. And we have to start with precision. The tonoplast. It is definitely not just a thin membrane. The tonoplast is this specific, partially permeable membrane surrounding the plant cell vacuole that controls what goes in and out. Using generic, run-of-the-mill textbook terms instead of this exact biological name is a notoriously fast way to just throw away points. You have to use the specific vocabulary designated by the 9700 syllabus.
So, why does ordinary revision fail here? Well, a low-band student might write, the plant cell vacuole is important for water balance. Immediately, the examiner's red alert flashes. It's vague terminology, and it shows confused cause and effect. Instead, high-mark strategies require you to link turgor pressure and the tonoplast permeability directly to the data you're given in the prompt. You actually have to explain that water moves down a water potential gradient, which establishes turgor pressure against the cell wall. It's a literal chain of events, and it's completely reliant on that partially permeable tonoplast.
To systematically deconstruct and solve literally any complex data question regarding plant cells, you really need to lean on a 4-step high-mark framework. Step 1 is concept identification. You have to identify the fundamental biological principle at play, like turgor pressure. Step 2 is evidence and data. Just extract those specific values from the tables or graphs they give you.
Step 3 is scientific reasoning. This is where you connect your evidence back to the biological concept using logical steps. And finally, Step 4, examiner wording. Translate your reasoning into the specific keywords you know are in the mark scheme. This framework is a game changer because it ensures you don't miss those critical method marks, even if your final conclusion ends up being slightly off.
Section 3. Cell membrane and cholesterol. Precision and evidence.
Let's actually apply this exact same framework to another really high-stakes topic. Imagine a flawed student answer that just says, the membrane changes, temperature makes it more fluid. I mean, that's going to get red pen everywhere. It completely lacks specific scientific reasoning and totally ignores data extraction. To get yourself into the top band, you need to say that an increase in temperature increases the kinetic energy of phospholipids, which then increases membrane permeability. And then, this is crucial, you provide a data value with units. Finally, you wrap it up by stating that cholesterol prevents the membrane from becoming too fluid. Boom! You've established a clear, evidence-based cause and effect.
So let's quickly summarize the specific dual roles of these key membrane components. Cholesterol doesn't just do things with temperature. It explicitly regulates membrane fluidity, preventing rigidity at low temperatures and excess fluidity at really high temperatures. Meanwhile, glycolipids and glycoproteins act as cell signaling receptors, recognition sites, and are absolutely essential for cell-to-cell adhesion. And remember, if a question gives you a graph about membrane permeability across different temperatures, you must extract the exact data values with their units to support your explanation of how these components work.
Section 4. The Interactive Exam Challenge. All right, you've seen the frameworks and we've talked about the red flags. Now I want to turn the tables for a second and challenge you. Imagine a student has written this deceptively simple answer on their exam. The nucleus contains DNA and controls the cell. I'll give you a second. Take a quick mental note. Why would this fail to get full marks from a Cambridge examiner? What exactly is the specific error here?
Well, if you said it was too generic, you hit the nail right on the head. Controls the cell is, quite frankly, a GCSE level answer. The error is that it's just way too vague. At the A-level standard, examiners demand precise, technical language. The correct phrasing you're looking for is, the nucleus regulates gene expression. You absolutely have to level up your vocabulary so it matches the syllabus requirements.
And this brings us to another really critical trap, the comparative analysis protocol. Whenever a question asks you to compare structures, say the rough ER and the smooth ER, you cannot just list what one has and the other doesn't. You must explicitly provide evidence for both similarities and differences if you want those top band marks. So a similarity is that both are membrane-bound organelle networks. The difference? Well, the rough ER has ribosomes for protein synthesis, while the smooth ER lacks ribosomes and synthesizes lipids. Explicitly stating both of those sides is completely non-negotiable.
Section 5. Final recap and strategy.
As we wrap up this explainer, let's consolidate everything into a basically bulletproof strategy for your next paper. If you only take away one thing today, let it be these top 3 examiner warnings, pulled straight from the principal reports. Number 1, vague descriptions will sink your grade. Always use syllabus-specific terminology like 70S ribosomes and tonoplast. Number 2, the missing units penalty is very real. Always check your scale and convert millimeters to micrometers when it's required. And number 3, context neglect. Please do not just regurgitate memorized facts. You have to tie your biological reasoning directly to the specific scenario or the data provided in the prompt.
I want to leave you with this final, genuinely critical question to ask yourself every single time you put pen to paper. Are you simply describing biological facts, or are you actually explaining the why with evidence-based reasoning? Shifting from just describing to explaining with evidence is the absolute key to A-level mastery. So keep this question in mind during your revision. Apply the frameworks we discussed today, and for sure, you'll be thinking exactly like an examiner in no time. Thank you so much for joining me on this explainer, and I really look forward to breaking down our next strategy with you soon.